So, welcome to the lecture on pentose phosphate pathway, also known as the hexose monophosphate shunt. Now, this week, what I made a small change to the schedule of teaching in terms of the fact that we will deal with glycogen metabolism next week because glycogen metabolism consists of two important pathways: one, synthesis of glycogen, which is glycogenesis, and the other one is the breakdown of glycogen, which is glycogenolysis. And these are quite elaborate pathways, and I wanted to keep this week's reading on the lighter side as you will be having the midterm exams. So, what I decided was to have a smaller lecture compared to the lectures on glycogen metabolism, which will extend a bit longer, such that you can devote more time to preparing for the examination. Now, the pentose phosphate pathway is a pathway that is not involved in the generation of ATP. Till now, we have looked at multiple pathways where the key focus was to look at how glucose is metabolized, and metabolism of glucose leads to formation of ATP, the energy currency of the cell. However, the pentose phosphate pathway is not involved in ATP generation, but it is also clinically a relevant pathway. So we have to focus on this pathway, but what we are going to do here is to focus on specific steps of the pathway rather than the whole pathway of the process. I come back to my old figure, which I think I introduced you to in the beginning of metabolism with regards to glucose metabolism. So, what we have done so far is that we have taken glucose and we have converted glucose to pyruvate, and the process that we talked about in quite a bit of detail was glycolysis, where we looked at the regulation of glycolysis, and then we moved on and studied the fate of pyruvate under aerobic or anaerobic conditions. And when we were talking about aerobic metabolism of pyruvate, we talked about the TCA cycle and the electron transport chain. We have also looked at the synthesis of glucose from non-carbohydrate precursor molecules, that is gluconeogenesis, talked about the Cori cycle and the glucose and alanine cycle, where I have introduced you to the working of the muscle, or more specifically, how muscle is able to generate ATP under different conditions of exercise and rest. So basically, this week and next week, we will focus on the other two arms of this figure. In this lecture, we talk about the pentose phosphate pathway, which is oxidation of glucose to ribose 5-phosphate. Glucose is a 6-carbon containing carbohydrate. Ribose 5-phosphate is a 5-carbon containing carbohydrate molecule. Next week, we will focus on converting glucose to glycogen, as well as we will look at how glycogen is converted to glucose. So that's like the summary of carbohydrate metabolism, which we will be able to conclude by the end of next week, or by the end of the week following your midterm exams. So today's lecture deals with glucose and its conversion to ribose 5-phosphate. The metabolic pathway that helps in doing so is the pentose phosphate pathway, also known as the hexose monophosphate shunt. So don't get confused if you come across this term, because they mean one and the same. We will follow the following roadmap. The most and the key take home message for you is what is the relevance of this particular metabolic pathway, and this I will like you to focus on while you are studying for your assessment, because this is a clinically relevant pathway when it comes about metabolizing glucose, and the significance of the pathway is not only important for your assessment at MBRU but elsewhere as well. As I told you, I'm not going to go through individual steps of the pathway. It is not required, and you may find it pretty complicated, but we will have a general overview for this pathway. And this year I have been able to come up with a small handy diagram that will help you to understand the general aspects of the metabolic process involved in this particular pathway. While we were looking at the pathway, we will divide the pathway in two phases, an oxidative phase and a non-oxidative phase. We will talk about the important aspects of these phases and then try to relate it to the significance or the relevance of the pathway. Most importantly, our focus will be on the committed step of the pathway and the formation of ribulose 5-phosphate. And lastly, we will talk about the clinical relevance of the pathway and also how to prepare for assessments when you are studying pentose phosphate pathway for the exam. So this is the general schematic of the pentose phosphate pathway. As I told you, at a glance it looks pretty complicated, although it is not so. So the starting material of the pathway is glucose, and the starting enzyme is all the time hexokinase, because as I told you when we were studying glycolysis, in order to trap glucose inside the cell, we need to phosphorylate it. So the starting point is always glucose, which will be converted by hexokinase to glucose 6-phosphate, and this particular conversion requires ATP. And once glucose 6-phosphate is formed, it then goes into different pathways depending on the metabolic state of the cell. And for the pentose phosphate pathway, the first intermediate that is formed is called 6-phosphoglucanolactone, which is ultimately converted to ribulose 5-phosphate, and ribulose 5-phosphate is then isomerized to ribose 5-phosphate. So I would like you to focus only on this particular part of the pathway, because this is what is required for you. The rest of the pathway involves rearrangements and conversions between 6-carbon and 4-carbon intermediates, which is not of significance for you, and you don't need to remember any of the intermediates or the enzymes except for the enzyme transketolase, which we will talk about quickly as we end the lecture. So if I want to have a figure which we can deal with very easily, I will focus on this particular figure. And if you see here, as I told you, the starting point of the pathway is glucose 6-phosphate. Of course, the starting point is always glucose, which is trapped by the conversion of glucose to glucose 6-phosphate by hexokinase. And glucose 6-phosphate is then converted to 6-phosphogluconate, and 6-phosphogluconate is converted to ribulose 5-phosphate, and ribulose 5-phosphate is then isomerized to ribose 5-phosphate. So glucose 6-phosphate is a 6-carbon containing carbohydrate intermediate, whereas ribose 5-phosphate is a 5-carbon containing intermediate. So pentose phosphate pathway therefore is a pathway that helps in the conversion of 6-carbon sugars to 5-carbon sugars. And why these 5-carbon sugars are so important is because of the fact that they form the backbone for the synthesis of DNA and RNA. So in case of DNA, we require ribose sugar, whereas in case of DNA, we require deoxyribose sugar, and both these types of riboses are derived from ribose 5-phosphate. In the process of conversion of glucose 6-phosphate to ribose 5-phosphate, you can see here at multiple steps you have oxidation happening, and NADP is converted to its protonated form, and this NADPH, that is the protonated form of NADP, is also important because it helps in the reduction of superoxide radicals in the red blood cells, and this particular process requires the participation of a molecule called glutathione. So the NADPH that is formed here is used not only for the regeneration of glutathione, but also it is used for synthesis of fatty acids and sterols. So the two key aspects of this pathway is to provide the pentose sugars for the synthesis of DNA and RNA, and also for the generation of protonated form of NADP, which is used in order to maintain the cellular integrity, especially of erythrocytes through glutathione, as well as the NADPH helps in the synthesis of fatty acids and sterols. So two important metabolites that we should focus on when we are studying pentose phosphate pathway is the generation of NADPH and the generation of ribose. So these are the two important functions of the pentose phosphate pathway. The conversion of glucose 6-phosphate to ribose 5-phosphate is an oxidative process, whereas the conversion of the 5-carbon intermediate ribulose 5-phosphate back to glucose 6-phosphate by the participation of the enzymes transketolase and transaldolase are involving the non-oxidative phase. So there are two phases in the pathway. One is the oxidative phase, the other one is the non-oxidative phase. If you are able to grasp these concepts, that is all that you will require from the pentose phosphate pathway. So you don't need to focus on any of the intermediates except the ones that are stated in the slide. So when you are studying pentose phosphate pathway, my suggestion to you will be to use this slide as a guide map in order to read through the concepts of pentose phosphate pathway. So as I mentioned, the pentose phosphate pathway is an alternative route for metabolism of glucose. It does not lead to formation of ATP, so it is not a process where you will generate energy, but it has two key functions. One is the formation of NADPH, and this NADPH is responsible for or used in the synthesis of fatty acids and sterols, as well as the maintenance of structural integrity of the RBCs through the participation of glutathione, which you observed in the earlier slide. The next most important function of this pathway is to generate 5-carbon sugars that in the form of ribose 5-phosphate, which is used for nucleotide and nucleic acid synthesis inside our body. So two key intermediates, NADPH and ribose 5-phosphate, are the ones that are generated from this particular metabolic pathway. This is not a pathway that is helping the body in the synthesis of ATP. So this is not a metabolic pathway associated with energy generation. Now, if I want to look at the summary of the pentose phosphate pathway, it looks something like this. Don't get confused by this quite a loud figure on this side. Just I will try to explain to you how this is working. So the pentose phosphate pathway actually starts with 3 molecules of glucose 6 phosphate. So you have 3 molecules of glucose 6 phosphate here on top. These are rearranged through a lot of steps, specifically involving isomerization. So if you look at the first key intermediate that you require from each glucose 6 phosphate, you are generating ribulose 5 phosphate, and this ribulose 5 phosphate, if you remember, the second slide is converted to ribose 5 phosphate, which is required for the generation or the formation of nucleic acids. In the process till here, we are also generating NADPH, which is the protonated form of NADP. And if you look at it, this NADPH is used for the synthesis of fatty acids and sterols, as well as to regulate the function of glutathione. So these are the two key aspects. Now, if you look at, as you go through the pathway, there are a lot of arrangements happening. So C5, C7, C6, and at the end you get back 2 molecules of glucose 6 phosphate and 1 molecule of glyceraldehyde 3 phosphate. Now, if you remember your glycolysis, you will remember that glyceraldehyde 3 phosphate is a glycolytic intermediate, but it has 3 carbons, so it will 2 glyceraldehyde 3 phosphate molecules will be required in order to generate glucose 6 phosphate back. That's the reason, since there is 1 glyceraldehyde 3 phosphate indicated in the figure, the figure states about half a molecule of glucose 6 phosphate. So it will lead to the production of glyceraldehyde 3 phosphate, and 2 molecules of glyceraldehyde 3 phosphate can regenerate glucose 6 phosphate back, and therefore the pathway actually accounts for the complete oxidation of glucose. So we start with glucose 6 phosphate, we get back glucose 6 phosphate by several rearrangements that happen in the process. And if you look at the important intermediates, NADPH, that I talked about, then we have ribulose 5 phosphate, which is converted to ribose 5 phosphate, and ribose 5 phosphate is important for the synthesis of nucleic acids. After that, the pathway continues with several rearrangements happening, leading to the formation of glucose 6 phosphate, another molecule of glucose 6 phosphate, and then we have glyceraldehyde 3 phosphate here, which then leads back to glucose 6 phosphate. And in the process, we will require 2 molecules of glyceraldehyde 3 phosphate. So that's the summary of the pentose phosphate pathway. We just need to focus on some of the key enzymes here. These enzymes, the other enzymes are enzymes such as the epimerase, ketoisomerase, 3-epimerase. We do not require to know about them. And also we do not require to know about the other intermediates of the pathway. So as I told you, this is exactly what you need to know, and that's the reason I have put this figure for you, so that you do not get confused when you are studying the pentose phosphate pathway. So let us now look at the various aspects of the pentose phosphate pathway. The enzymes of the pentose phosphate pathway are cytosolic. So the process, like glycolysis, occurs in the cytosol. Since it's a cytosolic process, that means even the RBCs will be able to carry out the pentose phosphate pathway, or any cell that lacks mitochondria will be able to carry out the pentose phosphate pathway. Now, the Key difference between glycolysis and pentose phosphate pathway is oxidation is achieved by dehydrogenation of NADP+. So we have NADP here, whereas in case of the other pathways like glycolysis, we have NAD+. So oxidation is achieved by the dehydrogenation using NADP+ as the hydrogen acceptor. So we protonate NADP, not NAD in pentose phosphate pathway. The full process of the pathway can be broadly classified into the oxidation phase, which is the conversion of glucose 6-phosphate to ribulose 5-phosphate, and a non-oxidation phase, which is the conversion of ribulose 5-phosphate by a series of reactions to glucose 6-phosphate. And two enzymes that are involved in this process are transketolase and transaldolase. Of course, one of those enzymes, as I told you, is clinically relevant for us, and we need to focus on this towards the end of the lecture. So let us now look at the committed or the rate limiting step of the pathway. So we start the pathway with glucose 6-phosphate. So before that, if you want to write when you are studying the pentose phosphate pathway, you can start with glucose, hexokinase, glucose 6-phosphate, and then the process starts from here. So glucose 6-phosphate is converted to 6-phosphoglucanolactone by the enzyme glucose 6-phosphate dehydrogenase. It requires cations as cofactors, mostly magnesium, and if magnesium is not available, the enzyme can also function in the presence of calcium. So in the process, the proton that is released, it is accepted by NADP+, which is converted to NADPH plus H+. So this particular enzyme is an NADP dependent enzyme, and I have written that in this part of the slide. After this, 6-phosphoglucanolactone undergoes a hydrolysis reaction. If you look at the process here, to 6-phosphogluconate, and this enzyme also requires magnesium and manganese as the cofactor, and also can function in the presence of calcium. So 6-phosphogluconate is the hydrolyzed product of 6-phosphoglucanolactone. So the committed step is catalyzed by glucose 6-phosphate dehydrogenase, NADP plus dependent enzyme, which converts glucose 6-phosphate to 6-phosphoglucanolactone. Once 6-phosphoglucanolactone is formed, it is hydrolyzed to 6-phosphogluconate by the enzyme hydrolase or glucanolactone hydrolase. So that's the subsequent reaction after the formation of the lactone. As I told you, we will have to look at the other step where NADP is protonated. So once we have 6-phosphogluconate, we have a second oxidation reaction that takes place, which is catalyzed by 6-phosphogluconate dehydrogenase, and this particular reaction also requires magnesium and manganese as the cofactor, and the enzyme can also function in the presence of calcium. The proton that is released reduces NADP to NADPH plus H+. So this is similar to what is happening in the first step. Just add a H here, which is missing. So this is what is the second oxidation step, and through an intermediate, this particular molecule, that is 6-phosphogluconate, is converted to the 5-carbon ribulose 5-phosphate. So if you look at the whole process together, you have 6 carbons here, and you have 5 carbons here. So you have been able to convert a 6 carbon molecule to a 5 carbon molecule. And when you are converting a 6 carbon molecule to a 5 carbon molecule, the carbon is lost in the form of carbon dioxide. So the conversion of 6-phosphogluconate to ribulose 5-phosphate is a decarboxylation reaction. And once this is over, ribulose 5-phosphate will be isomerized to ribose 5-phosphate, which is there in the second or the third slide of the lecture. And ribose 5-phosphate then through subsequent rearrangements is converted to glucose 6-phosphate, and one of the arms of the pathway produces glyceraldehyde 3-phosphate, which is also converted to glucose 6-phosphate. So you started with glucose 6-phosphate, you will end up with glucose 6-phosphate. In the process, you will also produce NADPH and ribose 5-phosphate. Both these intermediates, that is NADPH and ribose 5-phosphate, are important for the body in order to run other metabolic processes. So glucose 6-phosphate dehydrogenase is the rate limiting or the committed step of the pathway, and this enzyme is regulated by the availability of NADP plus. So when our body is actively synthesizing fats, then there will be a decrease in NADPH and an increase in NADP. So NADPH will be utilized in the synthetic processes or the pathways, and as NADPH is utilized, it will be converted to NADP. These increased concentrations of NADP will stimulate the pathway by allosterically activating glucose 6-phosphate dehydrogenase. Very simple regulatory mechanism. As we have more and more of NADP, we will have more glucose 6-phosphate dehydrogenase activity to replenish the NADPH stores in the body. And a major function of NADPH is to participate in synthetic pathways such as synthesis of fatty acids. Now, I kept on mentioning that there is a specific function for NADPH with regards to regulating glutathione activity. Now, glutathione, if you look at it here, you will see that glutathione is a small molecule, and by now you know that this is a disulfide bridge that is present in glutathione. Now, what glutathione does, since it's a disulfide bridge, glutathione has to be converted to a thiol group. And this process, that is the conversion of glutathione to reduced glutathione, that means in reduced glutathione, I am breaking the disulfide bridge and converting the SS to SH or a thiol group, involves the enzyme glutathione reductase. However, the proton required in the process is donated by NADPH. Now, the thing that you will be thinking is, why do we require glutathione? Now, inside the RBC, we have an environment which is rich in oxygen, and this leads to the production of superoxide radicals. Superoxide radicals have oxidative properties which may damage the erythrocytic cell membrane, affecting the integrity of the cell. So these superoxide radicals have to be neutralized, and the neutralization process is mediated by the reduced glutathione. So the reduced glutathione removes superoxide radicals in the form of H2O2 in a reaction catalyzed by glutathione peroxidase, and which contains the selenium analog of cysteine called selenocysteine. Now, what I'm going to do is I will describe the process here, but the aspect of selenocysteine we will talk during the lecture, because we will require to draw this out in order to explain it to you. So, for the time being, just look at the reduced glutathione, which reacts with superoxide radicals in the form of hydrogen peroxide in the presence of glutathione peroxidase, as a result of which water is produced and reduced glutathione is oxidized, so the thiol group is oxidized back to form the disulfide linkage. And I told you why this is important. The reaction is important since accumulation of H2O2, which generates superoxide radicals, may decrease the lifespan of the erythrocyte by causing oxidative damage to the cell membrane, which may lead to hemolysis. So, NADPH is important for the reduction of glutathione to form reduced glutathione in the presence of glutathione reductase. The superoxide radicals in the form of H2O2 then react with reduced glutathione in the presence of glutathione peroxidase to produce water and oxidized glutathione, which is again reduced by NADPH in the presence of glutathione peroxidase, and the cycle continues to maintain the homeostasis inside the red blood cell. So NADPH that is produced by the pentose phosphate pathway not only helps in the synthetic metabolic pathways, but also in the maintenance of homeostasis inside the cell. That brings us to one of the important genetic disorders associated with the pentose phosphate pathway, which is called G6PD deficiency. So some people have a deficiency of glucose 6 phosphate dehydrogenase, which attenuates the formation of NADPH. In turn, this particular cycle is deficient in these people, leading to hemolysis. So what I want you to do is to read about glucose 6 phosphate dehydrogenase deficiency from your textbook, and I am sure that we will discuss more about it in the tutorials on pentose phosphate pathway. So the regulation of the pentose phosphate pathway, I told you this. So this is kind of a chart that will help you to recap what I told you. So you have glucose 6 phosphate, 6-phosphoglucanolactone, and the pentose phosphates in the form of ribose and ribuloses. In the process, we produce NADPH. When NADPH levels are high, it inhibits the pathway by inhibiting glucose 6 phosphate dehydrogenase. When NADP levels are high, that means NADPH levels are low, it allosterically activates glucose 6 phosphate dehydrogenase, and this particular part of the pathway is favored. So this is also another aspect that I wrote here. So if you look at it, when glucose 6 phosphate enters glycolysis or the pentose phosphate pathway, whether glucose 6 phosphate enters glycolysis or the pentose phosphate pathway depends on the current needs of the cell, and on the concentration of NADP in the cytosol. Without the electron acceptor, the first reaction of the pentose phosphate pathway cannot proceed. When a cell is rapidly converting NADPH to NADP in biosynthetic reductions, the level of NADP rises, allosterically stimulating G6PD and thereby increasing the flux of glucose 6 phosphate through the pentose phosphate pathway. This is what is shown in this particular part of the figure. On the other hand, when the demand for NADPH slows, the level of NADP drops, the pentose phosphate pathway slows, and glucose 6 phosphate instead is fueled into glycolysis for the generation of ATP. Also, you need to look at this particular aspect, which is the requirement of NADPH in order to maintain homeostasis inside the erythrocytes. So you have the superoxide radical. The superoxide radical forms H2O2. This H2O2 has to be immediately neutralized. This is pursued by the enzyme glutathione peroxidase. We looked at this enzyme in the previous slide. In the process, reduced glutathione is oxidized. This oxidized glutathione has to be regenerated, and this is done so from the NADPH molecule that is generated from the pentose phosphate pathway. Once this oxidized glutathione is regenerated, it will prevent oxidative damage to lipids, proteins, and DNA. And most importantly, it is important for maintaining homeostasis inside the erythrocytes. Certain drugs such as ionizing radiation, sulfur drugs, herbicides, antimalarials, and chemical agents such as divicin, they increase the superoxide load in the cells. And if a person is deficient in glucose 6 phosphate dehydrogenase, then the person is also deficient in NADPH, as a result of which they may suffer from hemolysis. So this is one aspect that is clinically relevant for you, is to look at glucose 6 phosphate dehydrogenase deficiency. And I will talk about this in detail during the in-class session. But you should have a basic idea about glucose 6 phosphate dehydrogenase and its function in order to understand the importance of its deficiency. Before I end the lecture, I would like you to focus on a particular syndrome. I think we talked about this syndrome also a couple of times when we were talking about pyruvate dehydrogenase. And this particular syndrome is called the Wernicke-Korsakoff syndrome, sometimes known as the wet brain disease. And this is a particular syndrome that is observed in chronic alcoholics who are unable to absorb vitamin B1 or thiamine. And one of the reasons thiamine is required because it is activated to thiamine pyrophosphate, and thiamine pyrophosphate is an important cofactor for several enzymes. You know that pyruvate dehydrogenase requires thiamine pyrophosphate. Alpha ketoglutarate dehydrogenase requires thiamine pyrophosphate. But one of the enzymes of the pentose phosphate pathway also requires thiamine pyrophosphate, and this is the transketolase enzyme. So if thiamine pyrophosphate is deficient, the transketolase enzyme cannot function because it requires that as a cofactor. And I think when Mariam was teaching, she mentioned a mnemonic like ATP to remember the enzymes that require thiamine pyrophosphate. A for alpha ketoglutarate dehydrogenase, T for transketolase, and P for pyruvate dehydrogenase. So these are the three enzymes that essentially require TPP for their function. And if the person is deficient in thiamine, which can happen when the person is suffering from a disease called beriberi or Wernicke-Korsakoff syndrome for chronic alcoholics, then these enzymes have impaired activity. So what I would like you to do is to read this particular section from the slide, and it will give you a fair idea of what I told, not only now but as well as earlier in my sessions. So that's all. That brings us to the end of this lecture. I told you it's a short lecture just to focus on very specific steps, very specific enzymes. Next week we start, or next to next week we start with glycogen metabolism. With that, I will like to wish you all the best for your exams, and if you have any clarifications or questions, do not hesitate to communicate with me by email or over WhatsApp. Bye.